So once again, good morning, boys and Pallavi. And as I promised you yesterday, we are going ahead with eukaryotic cells in today's session. As we understand, eu means clear, well defined. Karyon means nucleus. So eukaryotic cells are those cells that have well defined nucleus. Yesterday's class we discussed about prokaryotic cell, in which the nucleus is not well defined, nuclear membrane is not there, membraneless cell organelles. So as far as evolution is concerned, they are very, very primitive. Whereas once the evolution has started, from prokaryotic, eukaryotic have developed, we see these changes. The first and foremost change that we observe is the presence of well-defined nucleus. I am sure in our biology lab, under the microscope, compound microscope, you have seen many cells, and the first and striking feature when you observe under microscope would be nucleus. You must have all noticed the nucleus, be it in amoeba, paramecium, or any plant tissue, epidermal tissue. I am very sure you must have observed the nucleus. So that is the best feature of eukaryotic cells. That's how the name also, eu means very clear, well defined, prominent. Karyon in Latin language is equal to, in English, nucleus. So eukaryotic cells are those cells in which there is a well defined nucleus, prominent nucleus with nuclear membrane. Okay, so they have well organized membrane bound nucleus as well as cell organelles. What are cell organelles? Parts of the cell, like all those structures that are present inside the cell: mitochondria, plastids, endoplasmic reticulum, nucleus itself, cytoplasm, all these are cell organelles. So presence of membrane in each of these gives clear compartmentalization of cytoplasm. It is just like you got a big hall, okay, and the big hall is divided into small rooms with some wooden structures. That's how in a cytoplasm we can see compartmentalization of each cell organelle, right? Now I'll just expand this, just see. They have complex locomotory and cytoskeletal structures. Locomotory structures that help in movements from one place to another place, it just moves. Okay, let me unlock that. Okay, right. Right. So the locomotory organs would be present that help in movement of the body. And cytoskeletal, cyto means cell, skeletal means the bone-like structures or some membrane-like structures. So these will give definite shape to the body. Imagine if we don't have skeleton, what would have been our shape? Thanks to the skeleton, the bones and the structures that attach bones to bones, I said the ligaments, bones to muscles, tendons. If these are not there, what would have been our shape? Just think of it. Without their presence, will it be possible for all of us to have the present shape that we are having? Impossible. Hence, the cytoskeletal structures also will give particular shape to the cell. And the genetic material is organized into chromosomes. So when we focus it under high power, all of you must have seen in your previous class, our compound microscope, the one that we are having in our lab, has got powers, high power and low power. So under high power, you would be able to see ribbon-like structures inside the nucleus, thread-like structures. Those are chromosomes. And all of you know, inside the chromosome there is genetic material in the form of DNA, RNA. and inside the DNA or portion of DNA is the gene. So if someone asks any one of you, what's a gene? You would be replying that gene is genetic material, secret of our life that is inbuilt in DNA. Okay. So it is the portion of DNA that is referred to as gene. So basically you have some chemical compounds. Those chemical compounds will make a gene, and genes portions together will make DNA and RNA. DNA and RNA together will make chromosomes. Chromosomes will be present in the nucleus. This is how the organization goes on. Now we have seen various cell organelles in prokaryotic cell. Let us see in eukaryotic cell. What are all the cell organelles that will be present? As we list, look at that. These are the organelles that we will see easily. Outermost, we will see the cell wall if it is a plant cell. Whereas if we see any animal cell including ours, there won't be cell wall. And just below the cell wall, there will be cell membrane, plasma membrane, which is made up of fats and proteins. We will talk about a lot about the chemicals present in the cell membrane or plasma membrane in this class. And then endomembrane system. Endo means inside, membrane having a layer. So there are structures which are having internally inside presence with prominent membrane. One of that is endoplasmic reticulum. We give abbreviation as ER. And in that two will be there, smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth endoplasmic reticulum will be just like one tube. Okay? And rough endoplasmic reticulum, on top of the tube-like structure there will be ribosomes. If ribosomes are present on the tube, then that tube we call it as RER, rough endoplasmic reticulum. Okay? Then comes again with tubes and round structures, that's called Golgi Apparatus. It is named after the scientist who discovered that, Golgi. And then again you will see spherical structures in the cytoplasm. They are called lysosomes. And yesterday I said a lot of questions will be there in your UPSC NDA as well as NEET exam about this. That there is a tag for every cell organelle. Like I said yesterday, mitochondria are the powerhouses of the cell. Why are they called powerhouses of the cell? Because they store energy. And the energy will be in the form of ATP, adenosine triphosphate. And we have got a tag for that. ATP is the currency of the cell, just like Indian currency is rupee, isn't it? American currency is dollar, American dollar we say. Just like that, our currency is chemical energy, ATP, and that ATP is present in the mitochondria. Since the currency is present inside the mitochondria, they are the powerhouses of the cell. Similarly, lysosomes are given a tag, suicidal cells of the suicidal bodies of the cell. Means they collect all the waste material, go to the surface area and blast themselves, break themselves. Since they are breaking themselves, it's almost like a suicide, atmahatya. Hence, all the waste materials will be thrown out by the lysosomes by blasting themselves off. And similarly, another round structures will be there. They are called vacuoles. And these vacuoles perform varieties of functions. Some vacuoles help in digestion. They are called digestive vacuoles. Some help in excretion. They are excretory vacuoles, like that. And then we can see, as I said, very prominent cell organelle, mitochondria. And normally the diagram will be asked of mitochondria. Then comes plastids. Plastids are of three types, in fact. Some of them already you know, very familiar, every day we are seeing in plants, that is chloroplast. Chloroplasts are the plastids that help in photosynthesis. All of you know in junior classes, particularly in 10th class, you studied light reaction, dark reaction, right, under photosynthesis. And there the sunlight is to be absorbed, and I am sure you must have studied that P1, P2 system, photosynthetic 1, photosynthetic 2. So that 760 nanometers or 700 nanometers wavelength sunlight is absorbed by chloroplast. And then second one is chromoplast. We are also seeing not only green color that is present in plants. Sometimes we are seeing varieties of colors in flower, in bud, in the whole plant, even in leaf. For example, crotons, you must observe a leaf with varieties of colors, red color, brown color, blue color, yellow color. So these colors are because of presence of chromoplasts. And then third one is leucoplasts. This is only storage. It stores foods. It is not responsible for any color, right? And these are also present in some of the lower animals. Whereas in our case, these will not be there. Why? We have got well-defined stomach, well-developed stomach. Then ribosomes, yesterday I told you, they are responsible for protein synthesis. Proteins are synthesized by ribosomes, and without proteins there will be no growth and development. Then comes cytoskeleton. Cyto means cell, skeleton, just now I said. There are some chemical compounds that will be present in the cytoplasm that will make the skeletal appearance. Then cilia, hair-like structures, flagella, whip-like structures. These are helpful in both movements, defense as well as offense. And then we have got centrosomes and centrioles. These help in cell division. We have two cell divisions already, all of you are familiar. One is mitosis, another is meiosis. And of course, we are talking of eukaryotic cells. Already I said eu means very clear, karyon means nucleus. So these cells have got well-defined nucleus, including our cells. And then there will be small, micro means small bodies. They are called microbodies. And now this is how a eukaryotic cell, a generalized structure would be. So all of you are required to draw this diagram. Soon after my class, when you are free today, you must draw this as colorful as possible, and preferably with pencil. No one will draw with pen, okay? And in our WhatsApp group, you will be posting a thumbs up, okay? Just put a thumbs up. That means I understand that you have practiced these two diagrams. This is the one, and second one also here you can see. These two diagrams I want you to practice. Okay, and these are there in your textbook also. And those of you who are not having textbook, I will post this. Please practice. Label it. Now, after labeling, I want you to understand the significance of each label. Whatever you are writing, like I read, let me go through that. First, you label nucleus. So write the importance of nucleus. Understand the importance of nucleus. Without nucleus, how the cell would behave. It is something like, nucleus is a controlling center. It's just like a teacher. How a teacher controls the class, nucleus controls all the activities of the cell. So first, after drawing, and remember, you all being biology students, you must draw most beautiful diagrams. You must impress the teacher, evaluator with your diagrams. And how do you do that? Just by practicing. First time when you draw, it looks very dirty, not good at all. Second time, third time, fourth time, maybe 10th time, it would be as good as the one that you are seeing. So practice, and practice makes not only men, Pallavi, women also perfect, right? So keep on practicing. And inside that spherical structure, nucleus, there will be another small round structure, that is nucleolus. Okay, nucleolus. Somebody is waiting, Shiv Kumar. Okay, right. So inside the nucleus there is a spherical structure, that is nucleolus. And what is the significance of that? Any idea? Anyone? What is the role of
It produces ribosomes.
nucleolus? Any idea?
It contains chromosomes, sir.
Okay, nucleus contains chromosomes. I am asking nucleolus. Inside the nucleus, one small TT ball like structure will be there. That is nucleolus.
It produces ribosomes.
No, ribosomes, nucleolus is not related to that. Okay.
DNA.
No. Okay, let me tell you. Nucleolus is responsible for cell division. If one cell has to become two cells, it is because of nucleolus, right? Next, Golgi apparatus. As I said, see, there are tube-like structures and spherical structures. Round bubbles will be there. So that is together. The tube-like structures and bubbles are Golgi apparatus. And the Golgi apparatus performs varieties of functions. One of the functions you must remember is it forms or it helps in formation of vacuoles. So vacuoles means again excretion. That means Golgi apparatus is responsible for excretion of the waste materials from the cell. Plus it produces lot many enzymes. You know, enzymes are responsible for our various activities like respiration, digestion, like that. And then since we are talking of eukaryotic cells, there has to be a nuclear envelope. Nuclear envelope is nothing but nuclear membrane. And then plasma membrane. Okay. And since we are talking of animal cell, remember, there will be plasma membrane or cell membrane will be the outermost. But this is, we are seeing, there is a label, outermost layer, down you can see cell wall. That means this cell that you are drawing or you are able to see is plant cell. I said plant cells only will have cell wall as the outermost membrane. We will be talking about this. What are the contents? What are the chemical compounds that will be present in cell wall? Why cell wall will be hard compared to plasma membrane? And plasma membrane or cell membrane is present just below the cell wall. Whereas in animal cell, including ours, there won't be cell wall. That is one of the fundamental differences between plant cell and animal cell. Okay? And then vacuole, look at that, the size of the vacuole. Almost it is 50% it is occupying of the cell size, isn't it? So if you see very big vacuole, big sized vacuole and less in numbers, you must understand that you are observing under microscope plant cell. See, just by observing closely, you will understand what type of cell you are seeing. Okay, somebody is waiting. Akash. Okay. Right, let me see whether the room is, it's unlocked room, right. Okay, so it is just by observing the cell closely, you can come to a conclusion that what type of cell are you seeing? Is it a plant cell or animal cell? How do you do that? First and foremost is nucleus. Of course, in both the plant cell as well as animal cell, nucleus is present. Nucleus will be present. But position of the nucleus. If the nucleus is pushed to one side of the cell, then you should understand it is plant cell. Who is pushing that? This big vacuole, big sized vacuole pushes the nucleus to one side. That means second thing that you must observe is vacuole. If there is very big vacuole which pushes the nucleus to one side, you must understand you are observing plant cell. Whereas, look at that, let me show you in animal cell. See in animal cell, Where is the nucleus? It's almost in the center of the cell. Why is it so? Vacuoles are small in size. There is no big vacuole that can push the nucleus to side. That is the reason why in animal cells the nucleus will be in the almost center of the cell. And third one you must observe is how many vacuoles are there, these big vacuoles? Very few. Very few vacuoles you will observe under plant cell. Whereas in animal cell, look at the vacuoles. They are small in size, their numbers will be more. So if you see these spherical structures, vacuoles in large numbers and small in size, nucleus is in the center, there is no cell wall, then you must understand that it is animal cell. I hope you get it that. And then we see the middle lamellae. The cell membrane will have 3 layers. So middle lamellae will be one of that. And just now I said cell wall is the outermost one. And then comes the mitochondria. Of course, singular mitochondrion, plural mitochondria, right? And there you can see the shape of that, and it's a double membranous one. Inside there will be finger-like structures, and I said mitochondria are the powerhouses of the cell. They are very, very powerful. Why? They have inside the currency, energy currency, ATP, and this supplies energy. Just like Raichur boys, they know that Shakti Nagar near Raichur will be is having rather electricity one. Raichur thermal power station, isn't it? That's why they are called Shakti Nagar people, and the area is called Shakti Nagar, right? And that supplies current, electricity to almost all the North Karnataka districts. So here we are calling mitochondria as powerhouses, Shakti, because of presence of ATP. Then comes ribosomes. Again, ribosomes are very small, spherical structures, and they are present in 2 places. One in the cytoplasm. Cytoplasm, liquid present inside the cell. Nucleoplasm, liquid present inside the nucleus. Plasm in Latin is liquid. So ribosomes will be floating. In fact, all the cell organelles will be floating in the cytoplasm. Ribosomes are small, round bodies. They are present in 2 places. One is, as I said, they will be floating in the cytoplasm, and the second one is they are present on the ER, Endoplasmic Reticulum. You can see, let me show you, yes. You can see there 2 tube-like structures. One is called smooth endoplasmic reticulum, another is called rough endoplasmic reticulum. And if you closely observe, smooth endoplasmic reticulum is having only a tube-like structure. Okay, the surface is very smooth. There are no any presence of any bodies on that. That's why we are calling it as smooth endoplasmic reticulum. Whereas if you see rough endoplasmic reticulum, let me expand that. Yeah, look at that. Rough endoplasmic reticulum. Are you able to see? There is a tube-like structure. Over the tube-like structure, you are seeing spherical bodies. Those spherical bodies, small spherical round bodies, are ribosomes. That means ribosomes, if they are present on the surface of the endoplasmic reticulum becomes rough. Hence, we have 2 types of endoplasmic reticulum. Smooth endoplasmic reticulum without ribosomes, rough endoplasmic reticulum with ribosomes. And that is the reason why I said that ribosomes will be present in 2 places. One is in the cytoplasm, floating freely. Another, they are present on the surface of the ER. ER means endoplasmic reticulum. Next, since we are talking of plant cell, chloroplast. Again, these are disc shaped, oval shaped. And I said the general name that we give is plastids. And just now I said plastids are of 3 types: chloroplast, chromoplast, leucoplast. And in plant cells we expect chloroplast, because the chloroplast will have a pigment, green colored pigment called chlorophyll. Sometimes in your junior classes your teachers must have asked why most of the plants are green in color. I am sure you must have replied that plants are green in color because of presence of chlorophyll, green colored pigment in them. And where is that chlorophyll present? In this chloroplast. And what is the property of that? It absorbs sunlight. By absorbing sunlight, chlorophyll is helping in plants making their own food through a process called photosynthesis. Photo means light, synthesis means preparation. Preparation of food with the help of sunlight. And what is the food of the plant? All of you know, starch. At the end of the photosynthesis process, what is being synthesized, what is being prepared is the starch, isn't it? And starch, all of you know, is a carbohydrate, and starch is a polymer. So chloroplasts are directly responsible for the preparation of starch, food. And next, cytoplasm. Just now I said cyto means cell, plasm means liquid. Liquid that is present inside the cell. And basically it is protoplasm. Proto means primitive, plasm means liquid. Liquid that is present inside the cell, and it is a primitive one, basic one. And this protoplasm becomes cytoplasm plus nucleoplasm. Nucleoplasm is liquid that is present inside the nucleus. So mathematically we can say protoplasm Pp is equal to cytoplasm Cp plus nucleoplasm Np. Okay, so Pp is equal to Cp plus Np. Next we have peroxisomes. Again they are internal structures. They help in production of various enzymes, that we will talk about that. Then we have got some small structures, they are called microtubules, we will be talking about them. And then plasmodesmata, there will be inside the plasma membrane some structures, we will talk about that. And lysosomes, I said they are suicidal bodies of the cell, atmahatya madkotavu. They kill themselves and throw the waste material out of the plant. So soon after the end of my class, draw this diagram, label this, and write the significance of each label. And one precaution that I would like to give you here is, whenever you are drawing diagram, arrow mark should be towards the part. Some of you have got the tendency to put arrow mark towards the name that you are writing, like lysosome. Arrow mark should be towards that round structure, spherical structure. That means pinpointedly you are doing it. Like in our biolab also, you must have used eyepiece or islands, and inside that islands there will be an arrow mark. So when you use that eyepiece, you are particularly pointing out one structure. You are not showing generalized one like that. And being science school students, you must maintain axis of the diagram. You must maintain what we say, just like Indian Army, single file. All of you are NCC cadets, senior division. Try to label on one side and don't do like Indian Air Force. So you label parties on left side, you put arrow mark and take it to right, or you put across the diagram one line. It appears as though you have cut the diagram. So don't do that. It should be on preferably one side. So I want you to practice this diagram, plant cell, and this today, animal. Okay. So I got a lot of time. These two diagrams I want you to practice. What is the difference here in between the plant cell and animal cell? As just by observing the diagram only you must have come to know. Talking from outside, periphery, Okay? You know that in plant cell there is cell wall, whereas here, search for cell wall. What is the outermost layer here? All of you are observing the diagram. What is the outermost one?
Cell membrane.
Cell membrane. Here we have labeled it as plasma membrane. Okay? And then microvilli, small structures. Golgi apparatus. Golgi apparatus or Golgi body, both are same. Smooth endoplasmic reticulum, nuclear envelope, nucleolus, nucleus, cytoplasm, rough endoplasmic reticulum, mitochondria, ribosomes, lysosomes, centriole, peroxisome. See, all these. So basically, when you observe both the diagrams, side by side you draw the diagram, you will come to know in plant cell outermost membrane is cell wall, as we have seen here. Look at that, cell wall. Whereas here in animal cell it is plasma membrane. Second, I said nucleus. Where are you drawing? In plant cell, just on one side of the cell you draw nucleus. Whereas in animal cell, draw centrally. Why? What is the reason? I just now told you. Presence of big sized vacuole in plant cell will push the nucleus to one of the sides. Whereas in animal cell, that is not the case, because there is no presence of big sized vacuoles. The vacuoles are very small. Look at that labeling where vacuole is there. Okay, centriole. Okay, you can label any one of the small round structure as vacuole. And the third one, in case of plant cell, draw one or two big sized vacuoles. Don't draw many. Whereas in animal cell, you can draw small sized round structures, many in number. So these are the basic things that you can draw. And the same thing we are writing the labeling: cell membrane, cell wall, endoplasmic reticulum, Golgi body or Golgi apparatus, lysosomes, vacuoles, mitochondria, plastids, ribosomes, and cytoskeleton, cilia, flagella, centrosome, and centrioles. Again, centrioles also help in cell division, mitosis, meiosis, and nucleus and microbodies. Microbodies, small structures present in that. So we will be going ahead with the cell membrane. Mostly in our next class we will talk about that. But again, I would like to show you the diagram, that is anatomy of cell membrane or plasma membrane. Okay. So chemical studies on human RBCs show that the cell membrane is composed of lipid layer. Lipid means fat. Okay, lipids are fats. Lipid bilayer, two layers of fats, and protein and carbohydrate. So if someone asks you what is plasma membrane made up of, your answer should be fats, proteins, carbohydrates. All three are present. And in that, in that lipids are two layered. And what type of lipids? Already in your chemistry class, your chemistry teacher must have told you varieties of lipids are there. Phosphoglycerides. So the lipids, being phosphoglycerides, have an outer polar head and inner hydrophobic tails. Now I am going to show you the diagram. Now before that, I would like to tell you what is this hydrophobia. Hydrous means water, phobia means fear of water. Like first time, remember when you were in 6th class in sainik school, new admission, your APTC fellow must have pushed you into the swimming pool. What was the fear you had? Almost you were crying and you thought that that is the last day of your life. But once you lost the fear, without the force from APTC fellow or your house captain, you were only jumping. And now you are fond of doing swimming. Isn't it? So if there is a fear of water, then it's called hydrophobia. And of course, the opposite, your chemistry teacher must have told you, hydrophily. Phily means love. Hydro means water, love for the water. You are fond of swimming, you are fond of water bodies. So, you have got phosphoglycerides which have outer polar head and inner hydrophobic tails. So, the non-polar tail of saturated hydrocarbons is protected from aqueous environment. Now, this is the structure how it will be. See, sugars are present, proteins are present, and two layers. Let me expand this. Yeah. Now, I think it is very clear. Two layers of lipid. Let me expand. Yeah. Now, look at that. That two layers of lipids. Bilipid. That's why we are saying. See, lipid bilayer. Are you able to see? That spring-like structures, not the round structures, okay? Spring-like structures. So, two layers are there, one up, one down. So, only bilipid. And then you have got protein molecules. We say integral protein means in between some protein molecules will be there. Even outside also they will be there. They are called external proteins. So, fat and protein are over. Now comes carbohydrate. Yeah, here we come. So, the protein structure, just now we said, tail and head. And look at the sugar molecule. To that protein structure, sugar molecule is attached. Sugar is a carbohydrate. And that is the reason why I said that plasma membrane chemically is made up of all the three components. Carbohydrate, that is sugar, protein with the two heads, okay, and third one is bilipid layer, two layers of fats. So, we will see again in our next class what exactly the structure of the protein, how it will be, right? Until then, I have given the task today. What is the task? You must draw both the diagrams, plant cell as well as animal cell. And then label it. While labeling, arrow mark towards the part. And try to label, I am saying try to label on one side. And then write the significance of each label that you have performed. And then in our WhatsApp group, put a thumbs up soon after completing the task. If you are not able to complete the task, means you are putting the thumbs up in our WhatsApp group. I would verify tonight how many have completed. Okay? Right. Take care. Bye. We'll see you tomorrow.
Sir.
Yeah. Yeah, tell me.
Sir, which unit produces ribosomes?
Ribosomes.
Which unit produces ribosomes?
Ribosomes are basically present in the cytoplasm. They are the parts of cell organelles. Okay? And they are responsible for protein synthesis. And basically everything is produced under the control of nucleus. Unless nucleus gives the orders, no cell organelle will be produced. Okay? And when we go in specific about ribosomes, I will be telling you details of ribosomes. Okay? Right. Take care. Bye.
Thank you.